Well, a very good day to everyone, and I hope everyone's had a terrific day so far. And thanks to everyone for being here. We're very excited to have everyone join us for our very first OzFlux Ecosystem Exchange webinar series. So it's all great to have you tuned in. And to start today, in keeping with the spirit of reconciliation, we acknowledge that traditional owners, that's the Noongar people of the Wadjuk lands here in UWA, where we are now. And we recognise that these have always been places of teaching and learning. Given that this is a webinar and we're in all different places across Australia and across the Tasman, even internationally, we acknowledge then the traditional owners of all the places and lands where we meet today. So we pay our respects to elders past, present and emerging, and acknowledge the important role that Aboriginal and Torres Strait Island people and other people have continued to play within our community. So again, thank you for joining us today. For those who haven't met me, my name's Jason Berenger and I'm the director of OzFlux. And for those of you who are not familiar with OzFlux, we are a facility within TURN, and we're an ecosystem research network set up to provide Australian, New Zealand and global ecosystem modelling communities with consistent observations of energy, carbon and water exchange between the atmosphere and key Australian and New Zealand ecosystems. So some practicalities for today's webinar. We cannot see or hear any of you as attendees, and so we will take questions through the Q&A box. At the top of your Zoom window, you should be able to see a Q&A button, and if you click on that, you can type your question in. So feel free to do that at any point during the seminar. However, we'll only be answering the questions at the end of the seminar. So we've got about 10 or 15 minutes at the end for questions. So we'll take those all at the end. And we also have a chat function for attendees. So if you want to use that as informal communication, do use the chat function as well. We've set up a very quick 10-second survey at the end of the webinar that'll appear in a separate browser. So once you've left the meeting, that will pop up, and we would really appreciate it if you could fill that in. Only take 10 seconds of your time to help our OzFlux reporting requirements. And we're also recording the webinar today so that we can share it with those of you who are not able to attend the webinar or want to watch it again. And the reason for having this seminar series really relates to the fantastic science that we do across the network. And so it's to highlight and showcase that science and the application of our work both here and abroad. We contribute to the global FluxNet community, and we thank all of the OzFlux community members and acknowledge the diversity of participating institutions, and in particular to TURN for their ongoing financial support. So without any further ado, it's my pleasure to introduce Dr. Caitlin Moore as today's speaker. And Caitlin completed her PhD at Monash University under glorious supervision, and that was on partitioning of the productivity and phenology dynamics of trees and grasses in Australian tropical savannas at the Howard Springs OzFlux site. So she's well accustomed to both extreme field conditions, but also with using flux data. Prior to this, she was employed at the University of Illinois, and after receiving an outstanding reference from her supervisors, she was gobbled up there at Illinois, and she was the first postdoc hired in the new Centre for Advanced Bioenergy and Bioproducts Innovation, where she was tasked with maintaining 6 existing flux towers. You're almost catching up to me, Caitlin, on that one. And she's worked on various projects, including water efficient sorghum. And her skills there are now perfect for her next project. The grass is probably not greener on the other side, but Caitlin has come back to Australia and across to WA. So she's now employed as a research fellow at the University of Western Australia on a collaborative turn project with WA partners, CSIRO, Edith Cowan University, UWA, Department of Biodiversity Conservation and Attractions. And her primary responsibility there is to install SIF and hyperspectral sensing capability. So she's also an invaluable member of the OzFlux Science Steering Committee. Today, she's going to be talking on her work from Illinois, and she's going to be talking on the bioenergy, crop fluxes, and an amazing conundrum. So Caitlin, I will hand it over to you.
Great, thanks Jason, thanks for that really generous introduction there. I'm just going to share my screen. Start this presentation. Okay, well, thanks for tuning in today, folks. It's really a pleasure to give the first of these, excuse me, OzFlux seminars. Today I'll be talking about, as Jason mentioned, bioenergy crop fluxes and an amazing conundrum. And this really represents a lot of the work that I carried out as a postdoctoral researcher in the Center for Advanced Bioenergy and Bioproducts Innovation at the University of Illinois. And so today, as Jason mentioned, I'm actually now a research fellow at the University of Western Australia. So I bring this talk to you from the University of Western Australia, which is based on the traditional land ownership of the Noongar people. But also the work was carried out in Illinois. And so I'd also like to recognize and acknowledge that the University of Illinois system carries out its mission on the namesake of a bunch of Native American landholder groups. And so the Midwest region, for those of you who maybe aren't familiar with the area, is indicated here in the US in green. So it includes a bunch of states that really are kind of oriented around the Great Lakes area. And Illinois is this one here that's circled in yellow. And it's basically characterized by fairly flat terrain with widespread corn and soybean fields dotted with little red barns across the landscape. Also famous for the Chicago Bulls and the Bear Cubs baseball team. The Blues Brothers, Abraham Lincoln ventured through Illinois quite a bit during his time, and deep dish pizza, which is basically like a pizza pie. So some fun Illinois facts for you there. And so to get to the heart of today's talk, why bioenergy? And so this figure here shows that in 2019, the US in particular, but this is kind of reflective of many nations around the world, have a heavy reliance of petroleum on providing transportation fuel. And while there is a renewable energy market occurring in the US, the biofuels only account for about 20% of this renewable energy contribution, which is only a small piece of the overall pie. And so with the good news that the US has now rejoined the Paris Climate Agreement, we're hoping to reduce this petroleum piece of the pie down by about 80%. And in order to achieve that, we can't just rely on electric vehicles. We also need to substitute that petroleum fuel with bioenergy. And so bioenergy has been projected to need to take up at least 50% of the slack in the reduction of petroleum fuel use. And so in order to achieve this, it's a pretty big task. So the US Department of Energy has funded several bioenergy research centers to really look at the sort of key themes on sustainability. So it's going to take quite a lot of land in order to produce enough biomass to be able to deliver these fuel emission target reductions for the future. And so coupled in that, there's also a climate challenge in being able to produce large amounts of biomass on the land that is actually allocated to growing these crops. But then also thinking about it in terms of biomass breakdown and deconstructing the biomass into the usable biofuel and bioproducts at the end. And so CABI is approaching this through three key research themes. And so we have a feedstock development theme, which is looking at developing more advanced crops in order to grow bigger plants with fewer resources. We're also looking at ways in which to convert these biomass product into more efficient and quicker to produce bioenergy fuels. And then we have a sustainability theme that's looking at not only the environmental, but also the economic feasibility and sustainability of an emerging biofuel industry. And so our scientific challenge overall, particularly from a sustainability standpoint, is that we're currently lacking an understanding of ecosystem processes related to the biogeochemical cycling of carbon and nitrogen and water in these different bioenergy crop systems. We also have a poor understanding of just how much land is available to actually grow and produce the bioenergy requirements for a bioenergy economy, as well as we need to understand a little bit better the refinery scale techno-economic analysis. And so how do we actually get biomass feedstock right through to a usable product? How do you distribute processing plants across a very large land area? Also a limited understanding, as I've mentioned previously, on the economic viability and resilience of a biomass industry. And so compounded on this as well, is that we've got a hotter and wetter climate forecast for the Midwest US. And so these states that are highlighted in green have been projected to experience hotter temperatures in future, as also more rainfall in these areas. And this can have pretty dire consequences for crop planting and crop growth. And so these two charts here are showing the consequence and the devastation from widespread high early growing season rainfall events that shows the difference in planting from May 2018 to May 2019, in that the planting was reduced because of widespread flooding. And so flooding not only waterlogs soils and makes it hard for the crops to emerge, but it can also damage storage infrastructure as indicated over here. And so by planting later in the season because of early growing season rainfall, it also shortens the window in which these plants have an ability to grow. These systems also, while hotter and wetter is the general sort of projection for these systems, they're also susceptible to drought. And so we've seen yield declines with hotter temperatures and drought already measured in the field. And so these panels here show gross primary productivity really starts to decline a lot faster during the year during a drought year, compared to more normal average climate years. And so in 2012, we had a widespread drought that occurred across the Midwest region. And you can see that the air temperatures were much hotter during the growing season. So days of the year from about 150 to 225. It's sort of the June, July, August time of the year. And you can also see that we had much less rainfall during these growing season months. And that then led to reduced yield from these systems. And so our CABI solution, I've got a number of points that we're working on within CABI and within the sustainability theme that I was part of to address some of these challenges. And so our hub and spoke model basically looks at core research sites and periphery sites to help examine crop performance under different genetic, environmental and management regimes. We also have very much a measurements and modeling approach to predict a better range of growth for these different bioenergy crop cultivars, as well as big data as well to quantify land availability. We're also including some techno-economic analysis and life cycle assessment studies to look at both the economic and ecological trade-offs of growing bioenergy in the United States. And looking also at identifying the optimal mix for like location of processing plants, where to grow the crops, marginal land areas that maybe don't compete necessarily with current food crop production, all in order to make a bioenergy economy more sustainable in the long run. And so here I'd like to introduce you to some of our bioenergy crop varieties that we grow at the Midwest. And so traditionally the land cover in the Midwest grows these high productivity C4 maize crops. It tends to be an annual crop rotation, so either a maize soy or a maize soybean alternation. And in the last sort of 10 to 20 years, the bioenergy crops that have been fairly widely studied in the Midwest region are miscanthus and switchgrass, and also mixed native prairie systems, because they produce a lot of biomass that can then be converted into bioenergy. And perennial systems tend to also promote soil organic carbon storage as well, because they add a lot of carbon to the soil as they grow. With cabbie, we've also introduced sorghum and sugarcane as new bioenergy crops, and these are really high producing, high productivity grasses. As indicated by myself, I'm 162 centimeters, standing there looking like a very tiny person up against this 5 meter tall sorghum, and it's so tall at times that you can lose a Mini Cooper in it quite easily. And so we're quite lucky at the University of Illinois to have a 320 acre bioenergy research farm located right in the heart of the high productivity hotspot area as indicated on this map. And so back in 2008, we had the establishment of the first flux towers at the Energy Farm research site. And so this included the conventional maize soy rotation, as well as miscanthus, switchgrass, and prairie as these perennial bioenergy crops that could be used as bioenergy candidate fuel sources. We also have a few other flux towers as well, which is feeding into the 6 that Jason mentioned that I've been looking after in my time at cabbie. And this is an intercropping sort of polyculture system, as well as duplicate maize and miscanthus plots for other research projects. And so using these 10 years of data, we can actually start to see patterns in which of these crops are sort of the winners in terms of their annual net ecosystem carbon uptake, as well as their water use. And you can see from this figure that the perennial crops, miscanthus, switchgrass, and prairie, are really starting to emerge as the winners in terms of, net carbon uptake, as well as their overall evapotranspiration. I'd also like to bring your attention to, in particular at the moment, to this switchgrass plot. And so after our 2012 drought, we can see that the switchgrass in green does start to decline in terms of its yield. And so at the time, in about 2013, 2014, we assessed that it actually had contracted a disease and which was affecting its vitality and productivity as a bioenergy crop. And so this led us to then develop a new study and test a hypothesis on what would actually happen to the carbon and nitrogen cycles if we were to revert our C4 perennial switchgrass field back to a conventional agricultural annual crop like maize. And so we got out there and basically tore up our 8-year-old switchgrass field and planted maize. And with our trusty flux towers in our field, so we had a control field, we had our historic switchgrass data, our control field, was maize, we had our historic switchgrass data, and then we planted maize in this converted field. And our flux towers then sort of began to tell us the story about what was happening to the carbon cycle. And you can really see here that there was quite a large shift in our net ecosystem exchange in the converted field, as indicated in the green. And when we partition these net fluxes into ecosystem respiration and gross primary productivity, we're starting to see here that the ecosystem respiration was much higher under the converted maize field. And when I say we, this work was very much a collaboration and made possible by my lab leader, Carl Bernacki, and my very talented technician, Evan Dracup. And so, given the fact that we've seen this elevated ecosystem respiration component, we also wanted to understand what components of the respiration were really driving this increased flux. And so we teamed up with Ilse Cantola and Mike Masters from Evan DeLucia's group, who looked at partitioning the autotrophic and the heterotrophic soil respiration components. And what we found was that carbon loss from the reverted field was driven primarily by higher heterotrophic respiration, significantly higher than the maize control field. And we found that this was most likely due to a higher below ground biomass left over from the switchgrass that existed previously. So there's a lot of biomass for the soil microbes to chew on to release higher CO2 from these systems. We also had a look at land reversion and how land reversion disrupted the nitrogen cycle, and found that particularly for nitrous oxide flux, our soil ammonium concentration, and our gross mineralization rates in 2017, that these were much more elevated in our maize converted field. And so this work was led by Rachel Van Allen from Wendy Yang's lab. And so our flux towers, is bringing you back to this net ecosystem exchange figure here. You can see that our control field our converted field, after 2 years really didn't, make it back, or didn't really reflect our control field after the 2 years of study. So in order to determine at what point did the system start to resemble a, conventionally tilled maize field, we enlisted the help of some modeling friends of ours, both at the University of Illinois and at the University of Idaho. And so this work was led by Danielle Berardi, who basically used the widely used DayCent model, which is a biogeochemical cycling model, and parameterized it. So we now call it the CABI DayCent model, because she parameterized it to include perennial crops such as switchgrass, so that we could then project out the time frame at which the converted field would lose all of the below ground carbon that had been stored from our 8-year switchgrass field. And this figure turned out to be about 5 years. And so we're also providing a similar approach to this switchgrass conversion down in Florida in the subtropics, to look at how conversion of pasture down there for the use of energy cane as a bioenergy crop can change the carbon, water and nitrogen cycles as well. And so the 2 flux towers pictured are towers that I helped to install, and they're now run by my friend Nuria, who's at the University of Illinois, and in collaboration with Betsy Borton at the Archbold Biological Station. And so part of my role on CABI as well was to install a new flux tower in a sorghum field. So our sorghum field is indicated here on the map at our energy farm in 2018. And this was mainly to get back at, like, with this sorghum field that's so big, how do the carbon, water and energy fluxes compare to the existing bioenergy crops that we're growing at these sites? And so preliminary work from our first year of measurement has shown that sorghum is, unsurprisingly, given the amount of biomass that it grows, a very high productivity annual crop, but it behaves more like a perennial in terms of its water use efficiency. And this is determined primarily by its gross primary productivity. And so you can see that this is daily smoothed gross primary productivity here in figure C down the bottom. And you can see that its GPP rate is more similar to a miscanthus, with maize outstripping both of them on a daily basis. However, maize also used more water during the process as well. And sorghum had a evapotranspiration profile much more similar to the miscanthus. And so when you combine these two to calculate a water use efficiency on a grams per millimeter basis, we see that sorghum actually performs best in terms of the amount of biomass produced per millimeter or litre of water used. Also, interestingly, we looked at the nitrogen flux profile as well. So cumulative nitrous oxide emission from these 3 systems. Not a lot really happening from miscanthus compared to sorghum and maize. And so this may be to do with this may be improved in sorghum in particular by better fertilizer application. And so my colleagues Mark Burnham and Adam von Hayden are really looking into this in future research as well, to really kind of optimize the fertilization rate of sorghum to lower this cumulative nitrous oxide curve. And so for me, I guess overall, how does this fit into the wider bioenergy sustainability picture? I think, having an understanding of ecosystem scale fluxes and how they differ or are similar between these different crop varieties is a really important step towards understanding the overall sustainability. And so I work in particular as part of an ecosystem measurements team quantifying these above and below ground fluxes, which then feeds into, informing our modeling team on the biogeochemical processes that are occurring, which then leads back into our 3 themes of CABI. And we kind of are really good at sharing what our findings and informing each other's research that way. And so that's sort of how I see the importance of our flux towers contributing to this overall bioenergy sustainability story. And so at this point, you might be saying, this is all fair and well, but what about this amazing conundrum you've been telling us all about? And so now I'm going to get into that. I'm going to bring your attention back to this figure here that I showed before about the sort of historic fluxes. And you might have thought, what in the heck is going up with your maize soy fluxes in 2014 and 2015? You weren't the only ones. If you did think this, I also thought the same thing. What's going on with my fluxes here? When we partition them as well, we can see that indeed, the system starts off as a net carbon sink in terms of net ecosystem exchange, but then partway through the season, it turns around and starts to show a bit of a shift, and the system becomes a net source of CO2 instead of a net sink right in the middle of the growing season. And partitioning of these fluxes in 2014 and 15 in particular shows that the systems are still highly productive in terms of GPP, but we have a really high contribution of ecosystem respiration. And so to me, this just seemed kind of crazy. How could ecosystem respiration be overtaking, gross photosynthesis during the peak of the growing season in our maize field, where maize is such a highly productive, high photosynthesis crop? And so my first thought was, well, some bad data must have made it through the QA/QC process. This is not possible. And so I went back and plotted here. I'm showing every 30-minute value for net ecosystem exchange that made it through our range checks and our quality control assessments. And you can see over time. And so you've got time on the x-axis and net ecosystem exchange on the y-axis. And you can see that for 2014 and 15, we're still capturing quite strong negative net ecosystem exchange from these systems, which indicates highly productive, a lot of photosynthesis going on in these crops. But we also see quite high positive net ecosystem exchange, indicating a lot of CO2 being then released from these systems. Interestingly too, a lot of sort of baseline capping of flux towers often tends to exclude data above 20 micromoles per meter squared, saying that it's outside of the range of normal ecosystem productivity, or what should be typical or expected. And if you can see that if we were to do that, exclude all data above 20 micromoles for this system in particular, that we'd be excluding not only a lot of data in 2014 and 2015, but a fair number of points outside in other years, which we consider to be our more normal or typical years for this site. And so I then thought, okay, well, nothing stands out glaringly obviously wrong in this figure. So what about where there's big data gaps? Is there any big data gaps where the system is particularly changing? And so what you're seeing here are all the data that have been QA/QC'd. So we've got the largest amount of gaps once we've removed data because of they didn't satisfy range checks or footprint exclusions and that sort of thing. And so the best way to interpret these figures is to look at, think of them as, on the x-axis, you've got time or hour of the day, and then on the y-axis, you've got day of the year. And so each 30-minute value is plotted based on hour of the day and day of the year. And so you get these kind of fingerprints of the different parameters over space and time, over time. And what we can basically see with these figures here is that we do have some large gaps. So anywhere you see white space is gaps in our data. But these gaps don't really occur when we're seeing these high fluxes in 2014 and 2015. And in particular, you can see here that at night, so in the nighttime, you're actually seeing yellow in these fingerprint plots in 2014 and 15, indicating high nighttime CO2 flux is occurring during these 2 years. And so big data gaps, if there were big data gaps occurring during these big transition periods, or when the system is changing, that would be bad because our models are then training to fill these data. but there's no sort of big transitions really popping up in these data. And so gap filling is likely not to be affected by large gaps when the system's changing. So then I thought, well, maybe it's a calibration error. Maybe there's something wrong with how the system, how the gas analyzer was calibrated during the 2014 and 15 period. And so this figure here is showing time over the x-axis and CO2 concentration measured by our gas analyzer on the y-axis. And you can see that there's been a couple of changes or updates in the version of our gas analyzer. So we've gone from a 7500 to a 7500 RS, but there is no obvious step change or any kind of shift that's occurring in these CO2 concentrations, other than the fact that baseline CO2 has gone up, but we know this is the case. So over here at the end of 2018-19, we've got CO2 just above 400 parts per million. And so nothing in particular, there's no kind of linear drift obviously evident in these in this plot. And I've also looked at it by year as well, and there's no evidence of a calibration error or linear drift occurring. And so then I thought, well, okay, maybe there's something wrong with the covariance between our vertical wind and our CO2 flux. And so I had a look at the co-spectra, so how basically the frequency distribution of eddies that are contributing to our CO2 and water vapor and energy flux at these sites. And so what you're seeing here, energy, CO2 and water and how it compares to wind. And essentially the take home from these figures is that the -4/3 gray line, all the curves should be basically following this trend for all, in order to tell us that the system is actually correctly capturing our vertical turbulent fluxes. And so if you saw some kind of deviation from this -4/3 trend, then that would be a warning. However, this is not really the case for 14 and 15. And so these plots are really telling us that our system was functioning correctly. A little bit of kickback over here in 17 and 18 with our energy flux, but our CO2 flux for the majority of 14, 15, 17 and 18 as our typical years, they all look very similar. And so it's unlikely to be an issue in the covariance between our wind and our CO2 flux. I then thought, okay, well, given it's a gas analyzer, are our water fluxes, which is coming from the same instrument, showing any abnormal patterns? And so here I'm really zooming in on our particularly problematic year, which was 2014. And you can see how water vapor flux is kind of going up and down based on the day. So it's kind of got this diurnal variability going on. Interestingly too, our net ecosystem exchange starts around 0 before the planting occurs at the start of May. And then you can see it starts off as kind of what you would expect with photosynthesis or net negative flux dominating the time series. But then the system starts to shift as we move on, and there's these kind of burst events occurring in our net ecosystem exchange time series. And the system also then, tapers back down to hover around 0 at the end of the growing season. And so if there was some kind of linear drift going on, you wouldn't expect the system to then correct itself or come back to hover around 0 at the end. So then I thought, okay, well, there's a nearby site, a Bonneville site, which is about 20 Ks from our energy farm site in Champaign. And so this is a site run by Carl and Tilden Myers, and it's a no-till agricultural site. And so there are differences in land management between the two sites, but at the same time, photosynthesis of a corn grown 20 Ks away should be fairly similar between two sites. And so we can see here, we've got Bonneville laid over our energy farm measurements, and we can see that it seems to be tracking the net ecosystem exchange, the negative NEE, seems to be tracking fairly well over our energy farm measurements, not so much at night though. And when we tease these data points out, so here we're looking at Bonneville on the x-axis and energy farm on the y-axis, we can see that our daytime net ecosystem exchange values match up quite well, whereas at night, our net ecosystem exchange values at the energy farm are pulling away from that one-to-one line. And this could be partly due to the no-till, but it could also be due to higher microbial activity, for example, at the site. And so then I thought, okay, well, are there any climate anomalies? Now that we know there's this difference going on between the Bonneville site and our site, is there anything in the climate to suggest that climate could be playing a part? And so on the left-hand side here, excuse me, I'm sorry, we've got mean monthly air temperature. And in particular, our drought is highlighted here in the magenta color. So our drought was really the hottest period in this, that our systems experienced in the past 10 years. But interestingly, 2014 was one of the cooler years. And so we can see that down here, it's you know, not cold by any means, but it's certainly a cooler growing season compared to 2012. I should also mention too that 2012 was a maize year, and then in 2013 we went into soybeans, so different crop. And then 2014 was the first iteration again of maize in our rotation. If we then have a look at cumulative annual rainfall, we can see here the drought. So we're seeing this kind of plateauing or flattening out of rainfall, particularly in the growing season. So this is why it was termed a drought for the Midwest. Those of us in Australia wouldn't really consider you know, annual rainfall of over 800 millimeters a drought, but you know, particularly a water deficit during the growing season when these systems are relying on high amounts of rainfall was pretty devastating for the crops. Interestingly too, we see that in 2014, and I've tried to highlight it here with the yellow arrows a bit more, but there's 3 or 4 kind of large rainfall events that are occurring in 2014. And so we see this particularly well in our soil moisture. And so these scatter plots are showing daytime and nighttime differences between the years for soil temperature, soil moisture, air temperature, and ground heat flux. And the key takeaway from these plots, because I know there's a lot of colourful dots to look at, is basically that during the day and night, of course, we've got much higher soil moisture, almost at field capacity in some cases in 2014 and 2015. So these systems had a lot of moisture available to them. So then I thought, okay, well, do our biomass measurements support the high GPP fluxes we saw? Because while we also saw high respiration, there was equally high gross primary productivity for at least the earlier part of the growing season. And so the figure on the left shows our below ground biomass, and I apologize, it's a little bit hard to see because we've got the perennial below ground biomass mixed in here as well, which is always higher than maize in these systems. But in particular, our yellow bar for maize in 2014 was actually the highest recorded below ground biomass across the 10-year period. So we had a lot of below ground biomass in our maize system. Also, we had almost our highest annual harvest from maize that year, other than our, you know, the first year of when these flux towers were installed. So really high biomass year, particularly when you compare it to the drought. I mean, in 2012, our maize yields were nearly as, well, equal to soybean in a good year. And so that's not good for maize growers. So I also then thought, okay, well, our climate seems to be playing a role. Could land management be playing a role? And so this table's a lot to take in. So I've mainly highlighted the key elements. Over here, we've got our 2012 drought, but also at the end of that growing season, we had lime applied to the field, so in September, to help neutralize pH. Then we went into a soybean year, and then in 2014, we were back onto a maize rotation. But you'll notice that we, in addition to applying nitrogen every single year, because maize needs a lot of nitrogen to produce its high yields, we also added potassium and phosphorus on 2 occasions, once before the planting and then once after planting. And so I guess after investigating all these avenues, and I've also looked into many other potential causes as well, but of course didn't have enough time in our 1-hour seminar to go into absolutely everything. I hope that I've sort of sparked your interest a little bit, and I ask you now the question, are these fluxes real? I've talked about whether any bad data made it through QA/QC, or whether there were gaps changing in the system, or any obvious changes in CO2 concentration, as well as poor calculation in our covariances, or whether water flux was impacted. And so all of these, for the first, options 1 to 5 here, I don't believe there is substantial evidence that any of these items could have been impacting our fluxes that badly. Likewise, our energy farm comparison to the nearby Bonneville site was good during the day, but poor at night. So indicating there's a difference in our respiration, in particular, our respiration fluxes at these sites. The climate was definitely different in 14 and 15. It was a little bit cooler, but most definitely wetter. Not cold, but certainly wetter. Our biomass and harvest measurements support our high fluxes. So we saw, over here, we can see our GPP is still really high during our 2014 and 2015 period. It's just that our respiration is also pretty high. And so there's also some evidence to suggest that land management may have also played a role. And so an amazing conundrum, perhaps, or a perfect cocktail of climate and management conditions that then led to a burst of soil microbial respiration. And so I have no way of testing this hypothesis since this is all kind of in the past, but certainly I can't find any evidence of a fault in the instrumentation that I have looked for. And so if anyone's got any ideas, certainly let me know, something that I haven't looked for. I'd be more than happy to hear your suggestions. And I guess my key take-home lessons for this is, know thy site, which is a very famous quote from our OzFlux community, as well as know thy instruments. But I'd also like to add that checking and scrutinizing thy data is also really important. Otherwise, we might miss these important, outlier ecosystem events from our data sets, particularly in these long timeframe data sets. And so with that, I'd like to thank all of the people who have helped keep the wheels rolling with this research. I was fortunate that these research sites were already running when I arrived, and I've been able to contribute into keeping them running in my time at Illinois. In particular, I'd like to make a special shout out to Peter Isaac and Cecilia Ewens from our OzFlux community for their continued support in getting me up and running with PyFlux Pro. All the data I showed you today from the Flux towers have been processed through PyFlux Pro, and the community in Illinois still use PyFlux Pro to this day. And so big shout out and big thanks to Peter Isaac and Cecilia Ewens for your help there. I'd also like to thank all the CABI partners and partner institutions, and especially thank you for all listening. Thank you to you all for listening, and I can take any questions now. I see there's been a few that have popped up in the chat, so I will finish with that. Thank you.
Thank you, Caitlin. An amazing talk. Let me start by saying I really appreciate the thoroughness that you went to in terms of investigating whether or not you thought that the data was real or not. I think that's an important lesson for all of us that are dealing with site level data. All of this tends to get washed out in a sense once it goes to sort of large scale databases that then feed into FluxNet databases and so forth. So I think it's really important that we do, as individual PIs, take very good care of the data before it then goes into those larger databases. So feel free, everyone, to post some questions. We don't have any open questions just yet. One from Elise in the chat. Sorry?
There are a couple in chat, yeah.
Yep. Okay. All right, so from Max, thanks, Caitlin, for an amazing presentation. Just a couple of questions. One, have you looked at any correlation between CO2 and methane and nitrous oxide fluxes during 2014, 2015?
I wish we had those data, is the short answer. Unfortunately, given the longevity of these sites and funding cycles, 2014 and 15 happened to be the 2 years that we didn't collect soil respiration measurements and didn't quantify these sort of trace gas fluxes. And so, again, our flux towers ticking along are what captured these events. And that's why a lot of this is really, a hypothesis for me, because I can't go back and retroactively test these things. I wish I had the methane and nitrous oxide fluxes, or even soil cores to do some soil incubation studies to look at what microbial communities were there during that time.
That was a suggestion I had for you, was whether or not you could even maybe post the event take some soil samples and do the lab incubations anyway. It's not ideal because the conditions of the soils have changed, but you might capture the same microbial community more or less.
Yeah, and also from my understanding as well, the microbial communities are quite different between a C3 and a C4 crop. And so particularly coming off the back of a drought as well in 2012, where a lot of the excess biomass, or the corn stover we call it, was, you know, put back into the field. Under hot, dry conditions, microbial activity is reduced. And so by the time we got back to the 2014 growing season, which was really wet, and there's a lot of biomass around for the microbes to chew on, whether or not there's some legacy effect as well as some land management effect going on there as well. Again, it's a lot of hypotheses and not a lot of ways to test them, unfortunately, but yeah, I suspect that's what is going on.
So the second part for Max's question, could have the maize roots exudates in those particular wet and cool years played a role in supporting high soil respiration rate?
Yeah, I think so. Again, no way to quantify it, but these are all very valid and likely hypotheses or explanations for what was going on. I've presented this work to various different labs in America, in the US as well. And a lot of the microbial folks sort of say, yes, it's more than likely your soil microbes are having a party there. It would be nice to have soil exudate or root exudate information as well, but part of the discussion, I think, for sure.
Yeah.
Once I write.
It's always easy to ask these questions in retrospect. So from Elise, wonderful presentation, Caitlin. I wonder if the ecosystem respiration could be moisture limited.
Yeah, yep. I think that's also playing into the mix as well, because we saw that in 2014 and 15, when we had the wetter growing seasons, and in particular, I think the timing of the wet makes a big difference as well, because the wet events were occurring in July, and in July, that's when our crops are at their peak productivity, but also the whole system is really at its peak productivity. And so when you've got these wetting events, it normally gets a little bit more drier in July in the Midwest. We get a lot of rain, as I showed early on. If you get it concentrated in April, May, when the planting's occurring, it can be detrimental to crop productivity. But certainly many rain events in the peak of the growing season in July, I think really would have fueled this enhanced microbial respiration. And it really, I mean, you saw that time series plot of, you know, the bars showing, you know, the large source, the positive net ecosystem exchange fluxes in 14 and 15. And it really shows how these systems can really be a burp of carbon from the ecosystem under ideal climate and or management conditions as well, because there's also, you know, the addition of extra fertilizer that I think would have also promoted enhanced microbial respiration.
All right, question from Lindsay in the Q&A. I'll just oscillate back between the chat and the Q&A questions. So Lindsay says, terrific talk, U-star and wind dynamics in the anomalous seasons, question mark. Did you see any ponding of soil environment during the wet periods? In effect, are your soils poorly drained and that amount of rainfall flipped the system?
So U-star I didn't include in this particular talk, but I have looked into it. The U-star thresholds were a little bit lower during 14 and 15, but not outside of the norm for some of the other years. We sort of have a range between about 0.1 to 0.25 U-star thresholds. And so our 2014 and 15 years were at the lower end of that range. But at the same time, would you say ponding as well, right? That can flip the system. And so I think particularly with the wetter growing season in 2014 and 15, I think that could have very well been the case. Yes, sorry, I lost my train of thought there a little bit, but but yes, I did look at U-star, and these systems, that tile drain, that's right, these systems are only rain-fed agricultural systems. So they do receive a lot of rainfall. I think annual rainfall in the Midwest is just a little over 1,000 mil, where we were in Champaign. And so the fields are actually drained. They've got tile drains installed to actually help, you know, remove a lot of this excess moisture, because these systems were originally prairie or wetland peat bogs as well. So they've all kind of been drained and cleared to make way for this high productivity agriculture.
Which perhaps relates to a question in the chat about how transferable are the findings to other sites, countries and crops?
I guess it depends on the system. I think for me, the key, I guess, the key warning or insight that this really highlights for me is that, you know, I think careful land management is really important, particularly if there's a high amount of biomass left over, or if there's been like a drought, for instance, where activity has been kind of fairly reduced as well. Because I think, and it's hard to tease apart the addition of, you know, good climatic growing season and the addition of the extra nutrients and the extra fertilizer from this system. And so, you know, maybe it's a case of, you know, we still had also bumper crops as well, right? And so in 2014 and 15, we had really high yields from these systems. But did that come at the cost of, you know, also expelling a lot of carbon from the system that really goes into long-term soil organic carbon storage? And so I think going forward, I think it does tell a bit of a land management story, particularly in the aftermath of any kind of, you know, climate stress event like a drought, for instance.
So a question from Dave Campbell. Was the ecosystem carbon balance compiled by taking account of other carbon inputs and exports as well as CO2, so that the change in soil carbon stock could be assessed?
Well, we've done it. We've calculated a net ecosystem carbon balance where we essentially subtract yield, the carbon content that's removed from the system in yield from these systems. And of course, we see an even bigger jump than we did with the net ecosystem exchange. These systems don't really, we've had methane sensors on these systems in the past and quantified methane emission. Evan DeLucia's group have led a lot of this work, and there's really not a lot in terms of methane that's emitted from these systems in the Midwest. Like in our systems down south in Florida, we get a lot more methane emissions, but not so much going on in the Midwest. But no, we don't have, I don't have any of the other more trace carbon fluxes to calculate a full, complete net ecosystem carbon balance other than removing carbon from harvested yield.
Yep, okay. A question from Atban, awesome presentation. The reason might be applying a significant amount of straw to the field. I wonder whether this could be checked out using some sort of remote sensing.
Yeah, that's a good question. I guess, I don't know how remote sensing necessarily would detect it, not being a remote sensing expert myself, but certainly with lower microbial activity, particularly in the drought, I think there was likely to be a lot of corn stover left over in the system, which wouldn't necessarily be consumed as heavily or as readily by the microbial community under a C3 soybean crop the next year. And so again, it's guessing whether or not a lot of that 2012 biomass was still in the system when the field was planted in maize back in 2014, so after the soybean year of 13. Again, it would be nice to have a crystal ball to go back and go, or maybe a TARDIS or something, go back in time and look. But yeah, I think for me, I guess the overall message is, these outlier events can actually, I mean, my thinking, I originally thought these data were incorrect when I first plotted them up, but I've been chewing on this for a while, and the more I look at it, the more I think about it, I think these fluxes are real. They've just captured this perfect cocktail.
Yeah, and given that your partitioning into GPP and into ecosystem respiration, the GPP part is more or less right because it matches with the biomass estimate. So if you get one half of the sum right, then you should be more or less be getting the other half right, or believe it.
Yep.
Gives you more confidence in that measurement anyway.
Yep.
Question from Chris. Thanks, Caitlin. That was very interesting. Did you notice any stress or die-off in the crops in 2014 that could have enhanced the respiration?
Anecdotally from people there, no. I wasn't at the University of Illinois in 14 and 15, but, you know, recollections of people who were there said, you know, all the comments were, yeah, it was a really, like, it wasn't super hot, but it was a super wet year. I'm not sure whether it was wet enough to be, you know, full like flood immersion of these cropping systems, or whether it just, you know, kept soil moisture at field capacity, or, you know, a ready supply of soil moisture for longer. I spoke with the farm manager, tried to pick his brain a fair bit once I saw these fluxes, and like, Tim, you've got to tell me, like, can you remember what this was like in 2014? And he doesn't recall any significant dieback or anything like that, or any particular, like, you know, he didn't notice any pests or disease that could have been consuming above ground biomass while the plants were, you know, highly photosynthetic. And so, yeah, sorry, I can't really provide a very good answer to that other than, yeah, I tried to dig in, but yeah, no one really recalls any particular visible damage in the systems.
All right, so we've got a few minutes left, and I noticed there are probably 5 or 6 questions, so we're not going to get through them all. So if you want to email Caitlin later on directly, I'll put up those contact details in a moment. But I guess as chair, I'll just ask maybe a couple of quick questions. One is, you talked about bioenergy, particularly in America, but I wonder, is there anything there that we could think about as an OzFlux network and as as to whether that's an aspect that we should be paying more attention to, or what's likely to be developing there? You mentioned sugarcane potentially as a bioenergy source. Is that something that we could be looking at as a network?
Yeah, I think there's certainly potential for even just for a bioenergy, or for some of these cropping varieties to become prominent in the bioenergy industry in Australia. We already have a sugarcane industry here for food purposes. But at the same time, I think a lot of the things we're learning from the CABI network about how efficiently these systems are growing. There's a lot of work, particularly in the feedstock theme of CABI that I mentioned earlier, going into making crops more climate resilient and able to be grown. I may have mentioned briefly this like marginal lands idea. And so there's a fairly large criticism at the moment that food crops, we don't want a bioenergy industry to displace food crops, because we also need to feed a growing population. And so there's a lot of research going into assessing these kind of marginal land areas where a crop like sorghum, for example, that is showing to be a more water efficient crop compared to maize, could be grown in drier areas where maize can't currently be, or isn't really viable even as a food crop. And so, I mean, Australia is the driest continent other than Antarctica in the planet. And so, you know, we could provide a really good test bed, or at least benefit from some of the advances that are occurring in terms of, you know, getting these crops to be more water efficient. And so some of that involves architectural modifications. So, you know, reducing the light intensity at the top of the canopy by changing leaf angle, as well as changing the amount and number of stomata on the leaves. So trying to reduce the number of pore openings of stomata at the top of the canopy, so as to reduce the amount of water that can escape from a canopy during the growing season, which is believed should be able to reduce the water requirements of some of these cropping systems. And so certainly, I mean, we should get a flux tower in a sugarcane system if we can over here, because I think that would really help to feed into it could even pair really nicely with, you know, the sugarcane tower that we've got recently set up in Florida. And, you know, do they compare well, or are they really different, and why? Could be a question that, you know, would be really valuable to the bioenergy industry.
Fantastic. Well, I'll save my last question for another time. And so I'd like you to all join me virtually to thank Caitlin for an awesome presentation. Thank you so much for taking the time to come and talk to us today, Caitlin. Her details are there on the screen. Email if you want to get in touch with her, or general queries through the Turn general email. You can reach people through that avenue as well. Our next seminar we're planning for May. We'll put out a doodle poll to find the best